This field is called as micrometry because of two purposes. One is like measuring small objects, measuring small objects or micro objects, and second is measuring through microscope. Now, obviously, when we need to measure the things, we need the scales. We need the scales, and not only the scale, but with proper units. Like our units should be very much defined. Now, we have spoken about the the limitations or the sizes of the parts of the cell. Many parts are like in nanometer. Many parts have the size of the micrometer. So, first of all, we need to come up with a scale that is fit enough, that is fit enough to permanently adjust in microscope. And the only way to adjust this type of object is one part that is called as the eyepiece. Eyepiece. The eyepiece is somewhat looks like this, and it has small opening over here, and then the upper part works as a different unit. It works as a different unit. Light reaches to this point, and your eye is over here, so you'll receive the light over here. Now, what happens that this part is removable. You can remove this particular part, and uh, then you have a small scale. This is the upper cap like part, this region, okay, this region. You have removed it, and now you have a very small scale. You have a very small scale that appears on the book like this, but it's almost like a very, you can say, the inside region of a ring.
Okay.
Like if you create a ring from that piece of scale, you can barely put it in your pinky finger. It
Okay.
has this much diameter. But for the sake of understanding, I am drawing it a bit bigger.
The
And
thing
we
that
have
we can, remove, what is it called? The one that you've drawn, that's the scale.
this scale
No, the red one that you would do with an
that you
arrow.
can see just the cap of the eyepiece.
Okay.
Now we remove the cap and we put this scale inside it. This scale has uh, this scale appears like this and so on, and it has 100 numbers.
This scale is already fitted inside that cap.
No, no, no. We put it inside.
Okay.
We put it inside. This scale is all easily purchasable. So
Mm.
we put it inside, and it has 100 numbers starting from 0 to 100.
Okay.
And then we put it inside, but this only has numbers. This
Yes.
has no units.
No units.
No units present on it. What did we say? We said that we should be having a scale that is permanently adjusted, but it should have proper units. This particular scale, it is called as eyepiece reticule. Eyepiece reticule. Mostly when we have a scale, we name it maybe a foot because it is composed of 12 inches. So it's a foot or it's a scale. So it is eyepiece reticule because right now it does not have any particular reading, any particular units. Now we need to give it the units. We need to give it the units. And how would we do that? we would be uh, we would be bringing another scale. This scale is rectangular in shape and almost as big as a microscopic slide. You can search it for the from the internet, that how much bigger it is compared to the eyepiece reticule. Microscopic slide, as big as the microscope slide. You can say I can draw it on the book, I'm sorry, on the sheet, um, but it is not that much bigger, and it has a very small scale inside here, and it might have some readings. It has some readings. When we magnify this particular scale already present in it, this is a prepared slide that we get from the market, from different shops. A prepared one. We do not have to prepare it. We just magnify it. When we magnify it, we have a whole large uh, scale. The scale starts from 0 point. Let me show you from here. It starts from 0. One large reading. One large reading is 0.1. And obviously, if it's 0.1, then the 10 readings within, then how much would it be? Zero.
Zero point zero
Every
one.
reading, yeah, every reading would be one small scale. Okay, sorry, let me correct it. One small scale equals to 0.01, and larger one equals to 0.1, and its readings are up to 0.25. 0.25. You can confirm it from the book over here. This is the last reading, and we need to calibrate. The proper word would be the calibrate. We
Okay.
need to calibrate which ones? The eyepiece reticule to this scale. This scale is called as stage micrometer. Now we did not name the previous one the micrometer. You remember? We did not name it the micrometer. Why? Because it's not in the measurements.
Yes.
It's not in the measurements, but now we have a scale that is in the measurements, and this is the stage micrometer. Now, the thing is, this is only for calibration. Only. We cannot put it above any specimen. This is
Yes.
totally wrong. We cannot put it above the specimen. So this is only for calibration. Now, how the calibration is done? Uh, you have eyepiece above. You put eyepiece over here, 0 to 100, and you have stage micrometer 0 to 0.25. Now, it seems that they are equal. Always remember that we magnified the things. We have magnified it. We have adjusted them to coincide with each other. To
Okay.
coincide. That is why they appear equal, but they are not equal. Now let's say that 100.
We place the stage micrometer below the specimen,
No, never, never, never.
above.
I
How do,
just
how
said
do we how do we coincide them? Where do we place the stage micrometer?
on the stage of the microscope, there is
Okay.
no specimen yet. There is no specimen. Right now,
Okay.
we are doing the calibration.
Only coinciding. Okay.
Yes. So,
Once we calibrate the eyepiece, then we can put the
specimen.
specimen. Okay,
Hmm.
done.
Now, 100 scale of eyepiece seems to be seems to become equal to 0.25 micrometer on the stage. 0.25 micrometer on the stage.
The stage micrometer
This is the
readings.
stage one.
The units are micrometers.
Yeah, obviously micrometers. So now, if 100 scales are equal to 0.25 micrometers, then what about the one scale? What about the one scale of eyepiece? If 100 scales of eyepiece are 0.25 micrometer on the stage, then what about the one scale of eyepiece? It would be 0.25 divided by
100.
100, and it would be 0.025 micrometer. Now we have our answer. One scale of eyepiece.
Can we say division instead of scale, or we need to use this word scale?
Um, I would prefer scale.
Okay.
Hmm. The calibration of the stage are 0.25 millimeter. The 0.25 millimeters is the unit. One scale equals to 100 scale 0.25 millimeter. One scale equals to 0.25 divided by 100. It would be 0.025 millimeters. Now we have one scale equals to 0.025 millimeter. Now we have to change the reading. Why? Because basically we are now entering in the
Microscopic
uh,
level.
yeah. So whenever we say when we say centimeter equals to 10 millimeters, and 1
Yes.
millimeter equals to 1000
1,000
micrometers.
micrometers.
So if we want to convert the millimeter into micrometer, so we would be multiplying it with the 1000. It's going to be 2.5 micrometer. So now you have one scale on eyepiece equals to 2.5 micrometer.
One scale on eyepiece, like each small division on eyepiece.
Each division on eyepiece.
Yes.
Okay, now how do we use it? That's the question.
Yes.
Let's say you have an organism under observation, and this organism is covering
Like
this
we
organism.
can you please
Mhm.
explain with the textbook? I think there is cheek cells there.
There might be cheek cells. Let's discuss a random example, then I can bring you there.
Okay. Okay, sir.
An organism, let's say that organism is covering, it's a bacteria,
Okay.
it's a bacterium, and it's covering, let's say, 10 scales. Now you remove the stage micrometer. Before moving ahead, remember that you have removed the stage micrometer.
Yes.
Once you calibrate it, the calibration is done. Remove it. Now you can put your specimens. Covering 10 scales of eyepiece. Okay.
Okay.
10 scales of eyepiece means you have to multiply it with 2.5, isn't it?
Yes.
Because you proved that one scale equals to 2.5. So if something is covering 10 scales multiplied with 2.5, so if we do the calculation, it would be 25.
Yes.
Hmm. It would be 25 micrometer. This particular organism that we just put under the microscope, under the eyepiece, it's 25 micrometer. Now, let's say these are the cheek cells. They are referring to this cell. This cell is covering 20 scales.
Sir,
So,
can you please zoom in?
okay.
Like from 40 to 60, right? That is how we assume
Yeah,
20.
yeah,
Okay.
from 40 to 60. So 20 into 2.5, what's going to be the answer?
For
20 into 2.5 is going to be 50.
yes.
Now, this cell is 50 micrometer in length, or you can say in size. So basically, this is how we do the micrometry.
Can you
Any
please?
question related to?
I got some idea. Can we practice two or three questions before the time ends from the past paper related only to this one?